Thank you. It is a real pleasure to be here. This is a topic that's close to my heart. So I'm an algal ecologist. I work on algal blooms. It's one of the main things I work on. And so it's near and dear to my heart. It's also near and dear to the heart of the organization that I work for, the International Institute for Sustainable Development's Experimental Lakes area. Because as I'm going to mention in a few minutes, that's why we exist. That's why the Experimental Lakes area was built in the 1960s. So this is a topic that's near and dear to my heart. And I'm really happy to talk to you folks all today and kind of really drill down to why it is that you're doing what you're doing. You know, why are you collecting these water samples for phosphorus? Why is it so important that you've been doing it? Why is it so important that you keep doing it? Right, so that's what I'm going to talk about. So we've all seen pictures like this. This was in the Free Press. A nice algal bloom washing up on the beaches. And for me, it's just a good reminder of why are we doing this? What's the ultimate goal here, right? Another picture. I find that these are disturbing, very beautiful photo, very disturbing when we get to the next slide that says, don't touch the water, don't swim in the water, don't let your pets go in the water, don't eat fish if they don't look like they're healthy. You know, these algal blooms can have real problems. Problems for the ecosystem, problems for human health and animal health. And, these signs are signs not to go on the beach or go in the water, but they're also signs that there's quite a big problem that needs to get fixed, right? So these are a good wake-up call for us whenever we see these signs that are on the beaches. But it's important to know that eutrophication isn't, or algal blooms, are not a local problem. This is not unique to Lake Winnipeg. This is probably the #1 leading water quality problem in the world. And that leads to what we call beneficial use impairments, or reasons why we can't use lakes in the ways that we would like to. I'd like to start with a bit of history about where we got to now, because we're going to talk about why phosphorus, why are we focusing on phosphorus. And if we go back to the 1950s and 1960s, this is a time when environmentalism was just really hitting its stride. Silent Spring by Rachel Carlson was published in 1952, just to put it in perspective. And there was a lot of advocacy by the public. People were getting really upset. And these are particular folks around the Great Lakes, large Great Lakes region. This is the time Lake Erie was getting major algal blooms, portions of Lake Ontario. Tributaries flowing into Lake Erie were catching on fire because of hydrocarbons that were released from industrial plants. And the public was really alarmed and really demanding that their politicians do something about this. But the problem was, there was a lot of scientific uncertainty. And the big question, at least in relation to the algal bloom ones, is what are the nutrients that were most important in controlling algal blooms in the Great Lakes and elsewhere? And there were 3. And if you collect a plant, or in this case algae, and you break it down into all of its parts, the 3 most things that are the most supply are carbon, nitrogen, and phosphorus. So these were the 3 elements that they said, okay, we think we need to focus. It's one of these 3 things that we need to focus on. However, at that time, and still today, science isn't the only thing that influences policy decisions. There's a lot of other considerations as well. And there was a very strong lobbying effort by industry. And at this time, one of those industries was the detergent industry. And at this time, there's a lot of phosphates in detergents, right? And so they were saying, there's a lot of scientific uncertainty here. We don't think that politicians should regulate phosphates because it just costs too much money, it would cost our industry too much, and there's a lot of uncertainty. And so you saw pictures like this one. We hung phosphates without a fair trial. And this was an industry article that was arguing against any policy action on this. Now, at this time, many limnologists did consider that phosphorus was the most important nutrient, the one that we needed to control and manage. But that wasn't the only voice that was being heard. There were a lot of laboratory and smaller scale experiments that were being conducted in universities and government labs that said, carbon and nitrogen can be limiting too. So there was a lot of uncertainty. As I said, the industry argued that the costs of regulating phosphates were just too high, and the science was equivocal. And so this led to inaction. A lot of algal blooms, nothing really being done. So this sounds very familiar to what we're experiencing right now. So what was the problem? Why did scientists and the scientific evidence not agree? What was going on? Why did this happen? And more importantly, it was cut off for those of you, is how do we arrive at a solution? How do we get there? And part of that is addressing that scientific uncertainty. So, this is supposed to say evidence-based policy and decision-making. And really what we want is at the far right side, we want a real-world solution. So, monitoring was showing that, hey, there's many lakes. Lake Erie was a problem at this time, but there's many lakes that have algal bloom problems, but the real-world solutions were still challenging, as we just mentioned. And the monitoring studies at that time did provide some evidence that phosphorus may be the element that's most important. And so you see here, this is phosphorus on this axis, and this is chlorophyll on this axis. And chlorophyll, of course, is that green algal pigment that's in all plants, and that we often use as an indicator of phytoplankton biomass. So this is really simple. The more phosphorus you have, the more algae you have. It goes up. Right? And so people would show correlations like this and say, okay, it really looks like it's phosphorus. But the problem is that you can also get very similar relationships to that with nitrogen. And so that's a very similar graph. This is, in fact, from the same lakes. Nitrogen's on the x-axis and algal biomass is on the y-axis. So there's a strong relationship. And what they pointed out, of course, is that correlation is not causation. All right, and so you can also do, I did this for fun, but this is pirate activity per year versus algal blooms. And you can get really strong relationships between things that have no relationship to each other. And so people argue, correlation is not causation. It doesn't mean one thing caused the other thing. And what it needs for, and this is the gold standard in science, is that you need controlled experiments in order to be sure. Right? Especially when you need to make policy decisions that are worth billions of dollars. You need scientific certainty. Okay, so here we are back again. Monitoring is great, and we need to do it in lakes. We need to track what's going on. But the problem is that there can be multiple stressors that are happening at the same time. We don't know which of them is having the effect. Correlation is not causation, they're not controlled experiments. So what else can we do? Well, there was two other types of experiments that were done frequently at the time. There's what we call lab or bench scale experiments. And these can be done I can do it in my laboratory, and my colleague at a university down the street can do it in their laboratory. So you can have a large number of experiments that are highly repeatable, and that's really good in science. And in these types of experiments, you would say you go maybe collect some water from your lake, you add a little bit of nitrogen to it, you see if you get more algae. You add a little bit of phosphorus, you see if you get more algae. And you add maybe nitrogen and phosphorus together, you see if you get even more algae. And those are the way those type of experiments happen. But of course, there are some problems with those types of experiments too. And the lab-based ones, often what they do, you take your water sample, but the first thing you do is you filter out any insects or invertebrates from it. So the grazers, things that eat algae, you filter them out right off the bat. You notice there's no fish in there for sure. They're way too small for that. But there's other components of the ecosystem that aren't there either. There's no sediments. There's no ecological feedback. So you really simplified the ecosystem down to just the algae and the lake water. Now you can get a little bit more realism if you go and do what they call enclosure studies. And these are like little bathtubs you put out in the lake, and it isolates a part of the water, and you can do the same sort of experiments you would do in the lab. But at least here, you have all those invertebrates, all those insects involved, you might have some sediments. But one of the problems with both of these types of experiments, they still suffer from a little lack of realism because they don't include the full ecosystem, but two, they tend to be quite short. The lab scale experiments tend to happen over days to a few weeks, and the enclosure experiments tend to last for weeks to maybe a few months, but almost always less than a year. So they're short. And so really this just summarizes it. So the criticisms were, those experiments are too short, they're too small, they're too simple, and they're not realistic enough. And the other problem, of course, is they gave conflicting results. Some of these experiments would say, hey, carbon's limiting, some would say nitrogen, some would say phosphorus. And this, of course, led to this policy in action that we got. So remember, we're back in the 1950s and 1960s here still. So politicians at that time said, we need something better in order for us to make these really challenging and complex policy decisions, things that are going to cost billions of dollars to enact. We need stronger scientific evidence. And I have to give credit to the politicians back then, because I don't know if they would do what this today, but they decided they were going to build the experimental lakes area. We need to conduct whole ecosystem experiments in order to solve this problem. And that's why the experimental lakes area even exists today. So we need experiments done at realistic scales to test the assumptions of these smaller scale experiments. And so that's what they did. In 1966, the Freshwater Institute, which is that large one of the buildings on the University of Manitoba campus, was built. They hired a number of researchers from North America and Europe, the real hotshots, and they got together, and some of them were working on the Laurentian Great Lakes, some of them came to Winnipeg and started the experimental lakes area. A few of the interesting ones, Wally Johnson was really important. He was one of the big visionaries, and he had previously gone to the University of Wisconsin-Madison, where they were also doing, in a smaller scale, whole ecosystem experiments. He said, this is what we need. This is what we really need to do to solve this problem. He brought on Jack Valentine, and Jack Valentine used to go to conferences wearing a globe on his back in order to remind scientists of why we're doing what we're doing. What's at stake here, right? So he was also known as Johnny Biosphere, was his nickname. And of course, you may have heard this name before. He's certainly popular at the Experimental Lakes area. His name is David Schindler. He was the first head scientist of ELA. And I laugh at, so this is Dave more, maybe a decade ago. But here's Dave when he's out of the site. And I chuckle at these photos because there's a few things you don't notice. One is life jackets. No life jackets. Two is shirts. It's such a neat shirt back then. And the other thing I'll point out, if you look where I've circled in red, that's phosphoric acid they're added to these things. They made them a lot tougher back then than we have now. I didn't add it, but I have a picture of me adding phosphoric acid to one of our experiments, and I'm covered in a suit. Every piece of skin is covered to make sure I don't get burned by the acid. They're a lot tougher back then. But this is what the experimental lakes area looked like back when it was first developed. It was actually just a bunch of construction trailers. They had ones for sleeping in, they had ones for kitchen facilities, and they had one for laboratories. And that was it. Get out there and let's solve this problem. And this is what it looks like today, much more established facility. And for those who don't know, it's most of the people who work at the Experimental Lakes area live here in Winnipeg, but our field station is just to the east of Lake Woods, right? And this is what it looks like. And this is our strategic plan, the main points of our strategic plan on the right. But there's I think it's the third one, and that's the most important one, and that is to support strong linkages between science and policy. This is what we exist for, and why we exist, and why I'm here today too. So this is really critical. We want a better future, and we want to try to base that on good science. The other thing I would say is just a little side note. When I was introduced, said my first real job was as a government scientist at the Experimental Lakes area. And I thought at that time, the way that management policy decisions get made is, I do my work out at my research, and I pass that research on to my manager, and somehow, through some secret method, it goes all the way up to the policymakers, and they make these great policies based on the work they did. And several years later, when I moved over to the nonprofit IISD, they laughed at me, but Scott, that's not how it works. And it works by the public being well informed and being motivated and telling politicians that things need to happen. Politicians don't really listen to scientists. They don't listen, and they often don't even listen to the managers, but they do listen to the public. And so this is another reason why I'm really excited to be here today and be joining the board as well. Now, several years, this is not in the 1960s, but Dave Schiller was reflecting on the importance of these large-scale experiments. And he wrote this, and I'll read it out so you don't have to read it in detail. The reason for the facility, this is the ELA, was very simple. Those responsible for managing large and important water bodies were reluctant to risk large sums of money on management schemes that have only been tested in short-term bottle or mesocausing experiments. Importantly, I believe that managers and politicians in today's conservative-oriented and economically-oriented, resource-hungry society are even harder to convince to act in an ecologically responsible manner than those 30 years ago, making it more necessary than ever to perform experiments at convincing scales. Now, Dave Schiller spent a lot of time meeting with politicians and trying to convince them to enact policy. He was called upon to testify in front of Congress many times and in front of our Canadian ministers. So he had experience. He knew what it took to get these policies moving. So the first question that they had to deal with then, and this is largely because of the detergent companies and industry, they said, how do you know it's not carbon that limits algal blooms? And it looks like my wonderful, I had this all perfectly laid out before, but they did have sort of a point. If you think of what algae need to grow, for every gram of phosphorus that algae need, they need 7 grams of nitrogen and a whopping 41 grams of carbon. So they need way more carbon than they need phosphorus. So surely, maybe it's carbon. Maybe that's what's causing all these problems. So the way that the experiment worked is they decided they needed to pick lakes with low amounts of carbon. And so these were lakes in the Canadian Shield that had low carbonate concentrations. That's why the ELA is situated where it is. And what you don't see below the way part is we're going to select one of those lakes, we're going to add nitrogen and phosphorus to it and see what happens. So this is the lake that they chose. It's not a big lake. You can canoe across it in about 5 minutes. It's 10 hectares in size. It's about 10 meters deep. It's pretty round for a Canadian Shield lake, pretty simple. And they added nitrogen and phosphorus to it, right? And the amounts that they would need for growth. I don't need any fancy graphs to show you what happened. Within the first year, they started to get algal blooms, and every year they added nitrogen and phosphorus to it, they got algal blooms. So year after year. So, solved. You don't need carbon. You don't need to manage carbon to limit algal blooms. That was the first question you got out of the way, right? But the politicians were still saying, yes, okay, that's great. But we need to know, is this phosphorus or is it not? Because that's what we're being asked to regulate, is phosphorus. So you need to do an experiment that really addresses this phosphorus question. So what they did is they chose a lake called Lake 226. All of them are numbered. We don't have any fancy names out there. And it is one of these more oddly shaped lakes on the Canadian Shield. It looks a bit like an hourglass. And what they did is they decided, let's take this double basin lake and let's split it down the middle with a plastic curtain, right? On one side, we'll add carbon and nitrogen. And you don't see it here, but this is a P for phosphorus. So on the other side, we add carbon, nitrogen, and phosphorus. All right, so let's see what happens. So here's this famous photo. And this photo is in all of the textbooks that we study as linology students in university. It's really clear. The side that got phosphorus had the algal bloom, and the side that didn't have phosphorus didn't have the algal bloom. This was the slam dunk home run that they were looking for, and policymakers were looking for, and it convinced them to move. All right? So, solved, maybe, phosphorus additions required for algal blooms. Right? So this is what led, or was an important contributor to, as the Great Lakes Water Quality Agreement around the Laurentian Great Lakes was being developed, that they could establish phosphorus controls within that agreement. They regulated for the first time phosphates in laundry detergents. They regulated phosphorus within wastewater treatment effluent for the first time. Right? And the establishment of tertiary treatment at wastewater treatment plants around the Great Lakes and eventually elsewhere. This is where we were in the 1960s. They didn't have these things, but this is what it took, these large-scale experiments. But notice, I had a question mark after solved, because it seems like you think you solved something, and someone kind of points something out to you. And what they pointed out is, well, yes, phosphorus was needed, but note that both nitrogen and phosphorus were added to that side of the lake. It wasn't just phosphorus alone. And so people started to ask that question, maybe we need to manage both nitrogen and phosphorus if we want to see a reduction in algal blooms. And okay, good question. And the other part was there's a lot of those smaller scale experiments that were saying the same thing. Many of those experiments say, hey, you know what, if I add phosphorus to one of these beakers, I might get a little bit more algae. But if I add both nitrogen and phosphorus, I get a lot more algae. So this really seems to me that we need to manage both nitrogen and phosphorus. And it isn't just the scientists that were arguing this. We have jurisdictions like in Manitoba that have decided that not only do you need to manage phosphorus, you need to manage nitrogen as well, right? And the quote below is for me, I believe, but scientists blast sewage treatment plants. So this scientific uncertainty leading to political inaction right here. So the question is a dual nutrient strategy needed? We also have some data from the experimental lakes area that might help with this. And it seems like they anticipated this question that we're having now, this real debate, because they said, we can keep adding phosphorus to this lake, but why don't we reduce the amount of nitrogen that we're adding and eventually just eliminate it altogether? All right, and let's see what happens. So here I'm actually showing some data. The experiment was in 3 phases. This first phase was where they added both nitrogen and phosphorus, and they added nitrogen with the amounts that plants need. Right, that's phase 1. Phase 2, they still added some nitrogen, but it was below the amount that plants generally need. So instead of 7 to 1, it's now being added as 4 to 1. Right? So this should be, plants can grow, it's giving them some nitrogen, but not enough. Did the size of the algal blooms go down? No, they didn't go down at all. In fact, there was one year where there was just an extremely large algal bloom. And then in phase 3 of the experiment that's existed since 1990, even though the data here stopped around 2012 or 2013, we're still doing this experiment. It's been going on for now 35 years. And the take-home is there's been no change. Right? No change in the size of the algal bloom. But this raises a really good question. It says, how do algae need nitrogen to grow? Where the heck are they getting it from? If they're not getting it from nitrate or ammonia that we have been adding into the lake, how do they get it? Well, it turns out, if you imagine you're looking through a microscope right now at a water sample that you took out of the lake, out of that lake, Lake 227. This is what you see. A little fuzzy too, but this is a filamentous algae that's actually been named after our first head scientist, Dave Schindler. So it's called the fence on one shingle. This is a real pinnacle if you get a species named after you. And this is filamentous algal blooms are common in when you have there's algal blooms. It's often filamentous algae, it's not always, but these are fairly common. And you'll see in here there's a few different things. Most of the cells are called vegetative cells. These are cells that are green, they photosynthesize, they grow, and as they grow, they divide, and the filament gets longer and longer. That's where most of the action happens. But you'll notice in this picture, there's a few other cells that look a little different. Those are called heterocysts, and they're unique to only certain types of blue green algae, and those that can fix atmospheric nitrogen. So these heterocysts provide an anoxic space where nitrogen fixation can occur. All of the other vegetative cells, they're photosynthesizing, they're producing oxygen. But it turns out in order to fix atmospheric nitrogen, that's N2 gas, you need this anoxic environment. So that's what those heterocysts provide. And I just want to point out, so we may not know it, but the room we're in now, the atmosphere, has about 70% of what we're breathing is N2 gas. That N2 gas just diffuses into the lake. It diffuses into Lake 227, it diffuses into Lake Winnipeg. And some algae, they don't need to use nitrate or ammonia. They just utilize that N2 gas. Right? And as we saw in the previous graph, they can use it, they do it just fine without nitrate or ammonia. They can fix enough nitrogen that it doesn't change the size of the algal bloom. And so that's what I'm showing here. This is the same figures I showed before, but the open symbols that you see there are now the amount of heterocysts that you find in water sample. So biomass doesn't change the size of the algal bloom, but we're getting more and more of those heterocysts in our samples saying there's more and more nitrogen fixation occurring in order to meet their nitrogen requirements. And so we were like, okay, we have now solved this problem. We know it's not nitrogen. We know we need to focus on phosphorus. But someone pointed out something, and they still said, maybe some of that nitrogen that you added way back in the first part of the experiment, maybe it's been recycling over time for 30 years in the lake, and maybe algae today are still using some of that nitrogen from 30 years ago. Okay, maybe. So we have a couple of modern experiments. This is a lake that we started work on in about 2018. And so we selected, there's two of these lakes we're doing this experiment on. We selected a couple of lakes without any history of nitrogen loading, right? We hadn't added nitrogen in the past. We're going to add phosphorus to them in the same concentrations that we added to that previous lake 227. No nitrogen additions whatsoever. And again, I don't need a fancy graph. That's what happened to the lake. It turned bright green. We started seeing algal blooms in the first year, and every year that we've been adding phosphorus to this lake, it developed algal blooms. And a picture I'm not showing here is we had to stop adding phosphorus for a few years during COVID, just because we couldn't get out to the site. Well, the algal blooms stopped, and we went back to having a clear lake again. Right? So it was pretty clear that this was a phosphorus issue. So we noticed there's no question mark anymore. At least that's what I feel. Some others, others may argue differently. But I think we've really nailed this one and said this is really clear. The other thing I wanted to point out in that previous experiment on Lake 227, where we slowly reduced phosphorus and then we stopped adding it altogether for now 35 years, we were able to remove 100% of that external phosphorus. So anything except rainfall and maybe what was coming off a very natural watershed. If we were to do this, say from wastewater treatment in Manitoba, we would not be able to achieve 100% nitrogen load reductions. We might be able to achieve 10% or 15 or 20%. But what this experiment shows is that the algae that are in the lake are more than capable of shifting over to these species that can fix atmospheric nitrogen and still create these very large blooms. Okay, so how do we, this is the most important part, how do we reduce algal blooms in Lake Winnipeg? So no big surprises here. and this is why, of course, the monitoring network is focused on phosphorus, is we need to reduce phosphorus load into the lake in order to reduce algal blooms. It's clear. The science is clear on this one. There's very little uncertainty. Now, there's been many years of inaction on phosphorus loading from wastewater in Manitoba for the same reasons that I talked about at the start of the presentation. Scientific uncertainty and leading to policy inaction. But we have certainty now, or as close as I think we're going to get to. And I was very pleased to note that, and this is pretty recent in the last year, and kudos to Alexis and the Lake Winnipeg Foundation team, is that strong efforts by the LWF and as efforts by the organization I work for, the IISD, helped secure interim phosphorus loading restrictions from wastewater treatment plants in Winnipeg, or the Northfield treatment plant. So this is very exciting. Finally, we may see that they start hitting the targets for phosphorus loads. But, and this is where you all come in, it's really important to know that most of the phosphorus that's coming into Lake Winnipeg is not coming from the city of Winnipeg. It's not coming from wastewater. 90% or more is coming from the watershed, right? And if we want to solve the problems of algal blooms in Lake Winnipeg, we need to address that problem. We need to address watershed loads. And unfortunately, climate change has had a role in making this problem worse. And Greg will be very familiar with, Greg McCullough will be very familiar with this graph. He helped develop this. This is a paper that he and Dave Schindler and Bob Heckey and some others wrote back in 2012. And there's a lot going on here, so I'm going to take you through it. Basically, it's saying that, back in the 1960s to the 1990s, here was the phosphorus concentration, the dark line that was in the Red River. Here's discharge, the smaller line. A lot of variables. Depends, is it a wet year, is it a dry year? And I highlighted this one because I'm interested in algae. So this is the phytoplankton, how much phytoplankton we have, and a sample from around 1970. Here's what happened around 1990. And this is the climate change angle. We started getting central Canada and eastern Canada started getting more wet. A lot more rainfall, a lot more total annual precipitation. So you saw Red River discharge go up, and it's really variable still, but it's reached a new sort of steady state at this higher level. Phosphorus concentrations went up in the Red River, and algae blooms quadrupled over that time. All right, and so there's this connection between how much rainfall, how much precipitation, snow and rain we're getting, and how much phosphorus loads are coming into the river, and how much algae we're having in the lake. And essentially, when I looked at this, I said, well, we can't control, at least in the short term here in Manitoba, we can't control this climate change. But what it means is this higher rainfall is making the lake even more sensitive to land use. So we have to pay even more attention to how we're managing the landscape than we would have had to back in the 1960s or 1970s, thanks to climate change, thanks to all this extra rainfall. And I would just like to finish off by saying, as you put this together, it basically says that your efforts, the monitoring network's efforts to track phosphorus loading in the watershed are what is critical to solving this problem. This is what we need to know. We need to know how much phosphorus is coming in. We need to know what the hot spots are. But the other thing we need to know is as we have that information and are really pushing the policymakers and the managers to make decisions and to try things out on the landscape, try out best management practices. We also need this monitoring data to let us know if those management practices were effective and how effective they were. So we need to track the effectiveness because then we can figure out what works and what doesn't, and we can apply that to other areas of the watershed. And of course, this can't be done without the data, and this can't be done without all the efforts to collect the data. So with that, I'd just like to say thank you very much, and happy to answer any questions if anyone has them.